Windmill. I'm just going to give 30 seconds so that we get everybody into the webinar, and then I'll make a start. Okay, so looks like the participants' numbers pretty much leveled out. So I'm Professor James Windmill. I'm going to give this webinar today on acoustic sensors inspired by insect ears. There will be time for questions at the end of the webinar. So if you put any questions into the Q&A, and I'll look at them when we get to the end of the talk part. Hopefully everybody can see the screen, no problem. So I wanted to just start with a little bit of my background, as why I'm here talking about this. I'm originally trained as an electronic engineer, then did a PhD in force microscopy, before then pivoting and taking 5 years of postdoc research looking at insect auditory systems. Looking for a full-time job, a permanent job, I then joined Strathclyde University in 2008 as a lecturer at the Centre for Ultrasonic Engineering, was then later promoted to professor, and then became director of the Centre of Ultrasonic Engineering back in 2021. Currently leading a centre with 11 academics and over 70 researchers. Personally, my own research activity is very multidisciplinary, looking at biological systems and then taking that and using it as inspiration for the conception and development of new sensor systems. And that's across a variety of different application areas, from consumer electronics, defense, medical to sustainable engineering. At Strathclyde, in the bioacoustics part of the Centre for Ultrasonic Engineering, we look at a variety of different things. So we do biological research, looking at insect auditory systems. We do a variety of advanced modeling, looking at those systems, but then also modeling to design new bio-inspired devices. All of that feeding into our bio-inspired sensor technology, which had looked at things like MEMS, microphones, ultrasonic NDE, 3D printed sensors, active acoustics, and then also most recently metamaterials. We used a variety of different techniques, which I wanted to cover at the start, so I'll refer to these later on. So we use 3D X-ray micro-CT. That gives us sub-micron 3D resolution of the thing we're looking at. And that can be looking at biological systems, but you can also be looking at things that we've manufactured to see how well they were manufactured. And of course, from that data, you can then create finite element models. Typically, we use COMSOL for that sort of modeling. And again, it's used on both our biological and our engineering research. On the experimental side, apart from, of course, measuring how things make sound or receive sound, we use a microscanning laser vibrometer. This is basically a way of measuring how much something moves. And for us, it's how much it's moving when you're playing sound at that structure, be it a biological structure or something that we've engineered. And so hopefully you're seeing the graphics at the bottom there, where the various different membranes of a MEMS microphone are moving up and down at different frequencies. That's not simulation, that's measured data from the vibrometer. So I wanted to start off by talking about directional hearing and miniature sensors. But why are they of interest to me, and why have we followed this research? So one of the first insects I worked on was the desert locust. You can see one sat there in the picture sitting on my finger. And then on the bottom left, you can see one with its wings cut back, and there you can see it has a temple and ear on either side of the body, which just sits above its hind leg. Now, these insects can hear roughly 300 hertz up to about 34, 35 kilohertz. But as you can see, they're relatively small. So what does that mean in terms of their directional hearing? Well, here's a COMSOL output looking at data capture from the X-ray system, and you can see the size of the insect's body. This is in relation to a sound wave at 1 kilohertz. And clearly, it'd be very difficult for that insect, even though it has ears on either side of its body to get any directional information. And so for the locust, it's only once we get up to higher frequencies, so up to tens of kilohertz, that you can see there with the sound wave that you're going to. That insect will then be able to get phase and amplitude cues, which will give it an idea of which direction the sound is coming from. For the locust, that's probably okay. Although it has this wide hearing range, it only really hears 3 or 4 different bands of frequencies. So at very low frequencies, it's only really interested in hearing, is it surrounded by other locusts or not? At the higher frequencies, it's probably interested on hearing whether there's a bat flying around emitting ultrasonic pulses, because then it can use its directionality to tell where that sound is coming from. However, some insects are much smaller and need to work at relatively lower frequencies. Now, this is going back to work done in the 1990s by my original postdoc supervisor, who looked at a fly called the Ormia. Now, this is the tiny fly. So in the image on the right, you have the big black thing is the cricket, and then sat on top of that is the little Ormia fly. That Ormia fly, the female searches for the cricket because it wants to put the larvae on the cricket, because the larvae will eat the cricket. It does that by tuning into the cricket's calling song. Crickets sing at about 5 kilohertz, and Ormias typically have a localization resolution of about 2 degrees at 5 kilohertz. The obvious thing that's of interest from an engineering point of view is that wavelength of sound in air is about 7 centimeters at 5 kilohertz, but Ormia's whole hearing system only spans, what, roughly 1.5 millimeter. So it can't be using arrival time in terms of sound pressure or difference in intensity. The size of the insect, the distance between its ears just don't work for that. So it uses some other trick. So on the right-hand side, we have a couple of images again from the X-ray system. And if you take off the head of that body, then underneath, basically on what we'd think of anthropomorphically as a chest, you have 2 tympanal membranes, fixed edge membranes. They cover the same airfield chamber. But the interesting thing with this insect is instead of just an air coupling, it actually has a mechanical bridge which goes across and connects those 2 tympanal membranes. And the result of that gives a very highly sensitive microscale directional acoustic sensor. Now, I'm not going to go into the very large amount of maths and physics and theory behind how this works. All you really need to know is that the way this thing moves gives 2 different distinctive modes, as I've tried to demonstrate there at the bottom. You have a rocking mode and you have a bending mode. And depending on where those modes are tuned in terms of frequency, you can get this system to give you a really nice directional output. However, there's one big problem with this. And so this is work done by you back in 2011. The Ormia is only interested in the 5 kilohertz that's produced by the cricket. It doesn't care about any other frequencies. And so it has this really nice tuned microscale system, which gives a really nice directional output, so long as you're looking for 5 kilohertz. At any other frequencies, then it really just doesn't work as well at all. Now, what's been done in the past, again, you're going back 20, 25 years, is people built MEMS versions of these microphones, but they tuned the modes such that you could get directionality across a broader bandwidth. The issue with that is that they were incredibly insensitive in terms of amplitude, so you need a very expensive method of measuring how that microphone was actually moving. In our group, we took a different approach. And we decided to look and say, can we make a microphone that actually responds at several different frequencies? So we use COMSOL to simulate what's happening with that microphone. You can get then the different eigenfrequencies and resonant modes, or you can do acoustic structure interaction. And so you can see here that we're getting 4 different frequencies. This happens because to start with, it's an asymmetric membrane. You can see it's rocking on that bridge in the middle for each of the images. But one side is actually slightly shorter than the other side. And then if you look closely, you can see that it's actually 2 membranes, one inserted inside the other, such that the higher frequencies, you're getting the inner membrane, which is doing the work and generating a directional response. And then those designs go off to a commercial multi-user process. They get made using piezo mumps. So it's a standard silicon device layer with some piezoelectric layer on top of that. You can also put in capacitive combs as a sensing method as well. And hopefully you can see here you have the outer membrane and the inner membrane, but they're asymmetric, such you then get 4 different resonance frequencies. One important thing which we found in this research, which was never really mentioned in any of the papers which we were reading, was you have to worry about how you're actually holding these things. If you just put them on top of your lab bench, then you get a very weird directional response. And so this is when we first got into 3D printing, actually making a holder for this. And that became important because if you look at these graphs, you can see in the blue and in the blue image, here you have basically a back cavity with air inlets. And in the red image, you have a back cavity which is closed off. If you close off that back cavity, then you get really bad directional response. You do not get anything like what you're expecting. Basically, you need to have the pressure release in the back of the cavity such that these things will work as directional microphones. But hopefully you can see, looking at the blue, roughly the simulation and the measured lines, the dotted and the solid lines, show that you're getting this bidirectional effect that you would expect at 4 different frequencies. And so we went on from that to say, well, can you try and get much lower frequencies? And so, yes, the answer is you can. So here we're looking at a microphone which has got, pardon me, 2 resonance frequencies, one just below 500 hertz and one just above 2 kilohertz. Again, this is with a device which is, say, 1 millimeter across and 3 millimeters long in terms of the membrane size. In this one, we're using piezoelectrics as well as capacitive combs as part of the sensing. And you can see in the image on the bottom right, there's a CR2032 coin-sized battery. And then next to that is the chip with the amplifier. And then you've got bottom, you've got the tiny chip with the microphone on it. Now, this is probably the best example of the issue with this type of directional microphone. The blue there is the simulation. The red is the measured. It doesn't look particularly pretty, but hopefully you can see with both that you're getting something which is working down around about 0.5 kilohertz and something which is working just up above 2 kilohertz. But in between and below and above, you're really not getting much signal, remembering that this is a nice log scale that we've got on the y-axis. These microphones are very good if you want to get directionality at particular narrow frequency bands. They're not that much good if you want to get a wideband response. But as you can see here, when you measure the directionality of that microphone for such a very small system, you get a really nice bidirectional system, which is at something like 200 times smaller than the actual wavelength of the sound. Our final bit of work that we did on this was looking and saying, well, okay, if you've got one microphone, then you're getting this backward and forward. What happens if you have 2 together? Rather than offsetting them, we actually built them into one system. So this is a system which is only working at one particular frequency. But you can see now that you've got 2 pads in one direction, 2 pads at 90 degrees to that. And hopefully you can see from the simulation and the measurement with the laser vibrometer there on the right-hand side that depending on which direction the sound is coming from, then a different part of that microphone will be innervated. So on the left-hand side here with the completed graph and the nice 3D graph, this was the simulations of the sort of output that you'd expect from that pair of microphones, so giving you a nice spatial 3-dimensional device. But on the right-hand side, that's the measurements that you're then getting. So basically, the PhD student measured one quadrant and said, yes, this is working. Again, this is the issue, though. It works really well at one frequency and not very well at any other frequencies. Now, because I've been involved with this a tiny bit, And because they get very grumpy if I don't mention it, I wanted to talk about much more recent work that other people have done on directional microphones. This is looking at using spider silk as the inspiration. So first of all, this is a talk about insects, and spiders aren't insects. And this is using the silk. So whilst the spider is using its organs to measure how the silk is moving, the actual microphone bit really is the web or the silk, and therefore it's really using a tool. The interesting thing here is because it's using a silk line or part of its web, then this is a measurement of velocities, kind of an airflow sensor, but the spider can use that to measure sound. As you can see there in the bottom right-hand side, sound wave hits the web, the spider picks it up. And this has now got to the point where people have made these and even commercialized them. So rather than silk, though, we're still using MEMS, we're still using silicon or similar microfabricated beams with a capacitive sensor, and you get a very nice directional microphone. Sensitivity now for these devices is similar at lower frequencies to standard MEMS microphones, and SoundScript over in North America have now commercialized this, and you can go and buy this microphone. The only one thing to say is because of the way it works with airflow and how it's designed, once you get up into even very low ultrasonic frequencies, then these systems don't work really at all. There's just no response. Whereas at least with the Ormia type design, you can get a response out of those. You can basically tune them up at higher frequencies if you'd want to take that sort of measurement. So Ormia, you can get a very tiny microphone compared to the wavelength. It's very highly directional. But the problem is, and has been for the last 25 years, people have been trying to make these, is they are frequency band limited. Very highly sensitive at that frequency band and can scale to a variety of different frequencies, including up into ultrasound. But you have an issue if you try to design this such you can get a wideband system. Much more recently, people have looked at the spider silk. You can get a good directionality out of that, a wideband sensitivity with a sensitivity equivalent to a standard MEMS device, but not suitable above 20 kilohertz. Now, what I'm not going to mention in this talk is that very, very recently, in the last year or so, there have been work has started to come out where people have combined these two together. And hopefully that's something we'll be able to read about in the future. Now, all of this has been MEMS hard silicon. And for me, about 10 years ago, I was thinking, this is nothing like the biological systems that I've worked on. I want to be able to make something which is not just inspired by the biology, but actually is in relation to the biology in terms of material properties, etc., having a three-dimensional structure. Obviously, the way that you can possibly think of doing this is looking at 3D printed systems. You can make three-dimensional structures. You can make things which have similar material properties to what you would find in the insects. And you can, to a degree, do them cost effectively. So we started looking at stereolithography, i.e. UV photopolymerization, relatively cheap, the sort of system that even 10 years ago, you could buy a nice system which gave you reasonably high resolution and is reasonably fast enough that you could use in the lab. Prices have come down in some aspects of that, such that you can actually imagine buying a system for your PhD student and letting them loose with it. Some of these systems are very nice because they let you have a completely open use of materials. So you blend the polymers that you want to use, and following on from that, that means you can functionalize them. So for example, if you want them to be conductive or piezoelectric or magnetic, there are ways of doping the polymer such that you can make that happen. So going way back when, obviously the first thing we wanted to do was say, well, we're interested in air-coupled microphones. Can we make a nice thin membrane which you could use for a microphone? So on the left-hand side there's the CAD. The very out of focus orange picture is what was then 3D printed. And on the right-hand side there, you can see the X-ray images. And you could quite easily print a membrane thickness of about 50 micrometers using a reasonably good quality 3D printer. And of course, once you can do that, then you can print things which are all sorts of different shapes and with lumps and bumps on them or different thicknesses. For us, the first thing we wanted to do was say, well, can we actually 3D print a microphone? Okay, we can 3D print a membrane that will move up and down, hopefully when sound is interacting with it, but we wanted something that you could make as a microphone. And so we looked at what sort of size do we need for the different resonance frequencies with the different material properties. So we ended up with a 3.5mm diameter diaphragm, did COMSOL simulations, did laser vibrometry of this. Of course, the COMSOL shown here is nice and really peaky because it's not taking into account the fact that the red is a true experiment. But then the really difficult bit was, how do you make this into a microphone? So you need a structure. You also need something which is going to convert what's happening, the force being applied on that structure, into an electrical signal. So you need something which is piezoelectric. So we use barium titanate as a composite with PEGDA. And then, of course, you need to measure that voltage. So you need the electrical part of this. So we used carbon nanotubes as a composite with PEGDA. And there in the image, you can see what we think is probably the world's first 3D printed microphone, a membrane thickness of 35 micrometers, sat there with a whole load of electronics because, unfortunately, it's a really, really bad microphone. But it did move up and down when you played sound at it. And if you look at the graph there on the bottom right, you can tell that this is a lab system with no shielding. You've got the EM signal there appearing from the speaker. And then just under a millisecond later, you get the signal appearing. The sound has traveled across from the speaker to the device, and the device is generating an electrical signal. So yes, you can completely 3D print a microphone if you really want to. That's me covering in a couple of slides what was probably about 2.5 years of work involving materials scientists, acoustics people, people who do the electronics, etc., etc. Following that, we also looked at capacitive 3D printed microphones. We cheated slightly with this in as much as one of the electrodes was actually a coating of aluminium that we put down. We didn't 3D print it. But we also used carbon nanotubes for the other electrode and a variety of different polymers in order to make the structure. You can see there in the bottom right, we did devices of various different diameters, and there it is sat on the preamp circuit. And there it is in a result, this time with 150 volt bias voltage. Blue is the 3D printed sensor. The weird greeny color is a B&K reference microphone. You can see that the reference microphone is very well shielded. Ours wasn't, because you get the input voltage to the speaker being mirrored on the capacitive sensor's electronics. But then about a couple of hundred, maybe a bit more milliseconds later, you can see that the sound arrives at both our 3D printed sensor and the B&K microphone, and they're both replicating it. Being a capacitive microphone, that you can play around with things like the bias voltage. You can see what sort of sensitivity you can get out of that and change the resonance a bit. But also then you could change the diameter of that microphone. And since we sit in the Center for Ultrasonic Engineering, that meant we could play around and see what sort of resonant frequency could we get out of that sort of system. We got it working up to eventually something like 80 kilohertz in air. So we can use a piezoelectric or capacitive system to build, 3D print some sort of acoustic sensor. But that then really needs to tie back to, well, the biologically inspired part. Now, roughly at the same time, we'd been doing work with colleagues looking at the lesser wax moth, or ochracea. The reason for this is the lesser wax moth generates its own ultrasound at around about 100 kilohertz as the male tries to attract the female. It does that by fanning its wings, which causes a click, which generates that sound. The really interesting part is that although this insect has 2 ears, as you can see in the picture on the right-hand side, which are slightly at a funny angle to each other, if you actually block one of those ears, then this insect still displays a directional hearing capability. In other words, it would appear from the biological measurements, basically playback of sound and seeing what the insect does, that even if it only has one ear, it can still directionally work out where that sound is coming from and follow it. And so that was a very much of interest to us. Looking at the structure, you have a thick part and a thin part. And if you look under the laser vibrometer, so this is a moth, if you play the sound directly down that center line of the black dots there at that ear, then you can see you get this big motion here of the bottom right, and that is motion where the neuron attaches to that membrane. So the membrane is rattling up and down in response to that sound if the sound is coming directly into that ear from 0 degrees. However, if you shift the sound around, you still get the same amount of motion, if that's the right way to put it, on your membrane. But you'll see now that the way the membrane's moving up and down is completely different. And what you see is that the part of the membrane which has the neuron attached to it, the bit which is the sensor, which then is the bit which is going to generate the nerve signal for the insect, that's not really being energized quite so much. If you actually then map it across all the different possible angles of sound coming into that ear in terms of intensity difference, then you can see that your moth with just one ear has a really nice directionality in terms of what it's getting out of that system. So why is that of interest to us? Well, that's a very small system. Now it's working up at 100 kilohertz. We don't need to make it quite so small. With the 3D printer, we can change our material properties, still make something relatively small, but we can print something which has got a thin part of membrane and a thick part of membrane. So on the top, there's a nice pretty picture of our moth, and the top polar plot, which looks quite bad, but it's because it's actual biological measurements which are difficult to do. That's the directionality that you get from the single ear of that moth. On the bottom left, there's a 3D printed moth-inspired microphone diaphragm. You can see it's roughly 2 millimeters across. One side is thick of that membrane, one side is thin, and you get the same sort of directionality pattern shown by the really nice polar plot there down in the middle at the bottom. The really nice thing about the moth system is that you can play around with the material properties and the scaling slightly, such that you can get this system on the 3D printed side to work down at lower frequencies. And as you can see, compared to the Ormia system, you can get a much wider bandwidth. Later versions of this, which I'm afraid I don't have a slide for, were getting down towards 1 kilohertz in terms of operating frequency and giving you a nice few kilohertz. And this is completely work which is not optimized. But you can see on the right-hand side that at different frequencies, you're getting a nice bidirectional pattern, both in a simulation on the left polar plots, and on the right-hand side, you have the experimental measurements, which generally tend to match that. Unfortunately, this work pretty much coincided with when we had COVID and the lockdowns and our laboratory being closed for probably about 18 months of this PhD student's work. And so they really weren't able to take this much further. But the plan is that we will carry on this work over the next couple of years and see how far we can get in terms of optimization. Now, we can 3D print systems. Another thing that you can 3D print or imagine 3D printing is something which is more hair-like. Now, people have been looking at insect hair systems for a long time as inspiration for sensors. Typically, insect hairs react to things like airflow or low frequency sound, frequency sensitivity being regulated by shape, size of the hair, stiffness of the socket. They tend to be directional. They can be used by the insect for other things like gravity, acceleration, or touch. Spiders use these, various other animals as well. And I'd also say we're not the first people to look at this. People have done a lot of work on this in the past, looking at inspiration from these using MEMS. but that's then MEMS typically using at least silicon as the base. And so various issues that I've already talked about in terms of material properties, etc. Material properties is what made us interested in this, because relatively recent work has shown that it's not just a single material system. Very often you'll find that the actual hair shaft, the bit that sticks up into your airflow, is very stiff. It doesn't bend, but it's attached to a much softer deforming area, which is where the sensor bit is. And there's usually a very high aspect ratio difference between these areas. And if you look at the Young's modulus, then people have shown that it can be up to 20 times difference between the nice stiff hair shaft, which is the bit which is going to catch the energy of the airflow, pardon me, and the springy bit, which is directing the energy then into where you've got your sensory neurons. Now we're using 3D printing, and so we can look at designs where you can imagine you can have a hair which has got nice and stiff. You can use a commercial resin, you get a relatively high Young's modulus, but then using one of our custom-made resins, you can make a much softer base spring. And of course, if you can do that, then you can start to imagine a design which is more closely related to this bio-inspired system. The only problem that we're kind of facing, given there's a PhD student looking at this, is that then you've got a huge different number of variables which you can look at. Just here's some of the work that he's done. So looking at different shape of hair, different height of base, how big is your spring base, how big is your actual hair, as in how high it is. And you can see, looking at the results at the bottom, that by playing around with all these different variables, you can move the resonant frequency that this system is working out up and down, and change the sensitivity, etc. Now, these are purely passive printed systems, the soft base, the nice stiff top. We've then started to do work over the last 6 months, maybe a bit longer, looking at how do you get an electronic signal out of this. And so the initial work was coating with silver, making a capacitive sensor, and you can get an output signal from that. But it's, at the moment, it's really bad. And so work is ongoing on getting that coating better, or maybe looking at something like a piezoelectric sensor, looking at how we can get a better signal out of that. But also the student's been doing some work with one of our colleagues in France, looking at particle image velocimetry to see, well, what happens when you change hair shape, etc. Can you get some guiding rules which will help guide the way that we take this work in the future? So our 3D printed microscale insect-inspired systems, you can 3D print a complete microphone if you really want to. You can make it piezoelectric or capacitive. You can 3D print with multiple materials. You can 3D print functional materials. You can make complex geometries. The main thing to say is it's really not that easy. It's me talking about it over a few slides, which is covering several years' work. But that has now led into the moth-inspired directional microphone, which has given us good directionality, relatively wideband sensitivity, but which is also scalable across frequencies. So we can put it at whatever frequency we're interested in, including up into low ultrasound. And then also we've been doing work looking at the hair sensilla-inspired sensor, typically looking at low frequency sound or airflow, but you could use it for gravity or acceleration instead. It's a directional sensor, and again, we can see that it can be scalable with frequency. Now, just for the last part of the webinar, I wanted to talk, and so I'm really reaching back to very early work that I did now, looking at active insect systems, but then that will lead back into where we're going with microscale sensors. So this is a moth which I was looking at when I was a lot younger, had more hair, a lot less gray hair. You can see that the same as with the other moth I spoke about on the bottom image on the left there, that's its ear. You have the white thicker membrane. Next to that is the almost transparent, thinner membrane. And then the arrow there's pointing to where the neurons attached to that particular membrane. And if you put a laser vibrometer on that, you can see it moves up and down. You get a lot of motion at the point where the neuron is attached. At lower frequencies, the big fat membrane is also up to something as well. It definitely doesn't move anything like a classical drum membrane. It's got masses and springs doing who knows what's on it. The really interesting thing is if you look at what happens in time, now in the blue graph, that's me playing pulses of sound at 30 kilohertz, 70 dB SPL at that ear. And the red is the response from the laser vibrometer. And as you can see, basically, hopefully, if you've got sound, you hopefully hear this. This is what I think I'm playing at it. But in terms of what the moth's neuron is getting in as an amplitude, it gets something more like this. So what's happening? Basically, the moth's tympanal membrane is changing its tuning. And so here on the looking at it in a frequency domain, the green part of that graph is showing what happens when you have very low level sound and you've got the moth tympanal ear tuned at relatively low frequency. Play lots of loud sound at it, and that tympanal membrane tunes up to a much higher resonant frequency. The gray background stuff is when the insect is dead or knocked out by CO2, and it just sits at a very low frequency and flops about a bit. And so you can actually see that that resonant frequency changes as you change the relative sound level. And it's actually a correspondence with energy. So in other words, you can keep playing the same frequency at it, and you see the frequency tuning changing with time as more energy has been put into the system. And then it will sit there and then slowly drift back down again. At the same time as we were doing this, one of my colleagues was looking at mosquito hearing. And this is a male tiger mosquito. He's interested in hearing the flight sound of a female mosquito. And if you look at the graph, there, then you see that you have the white curve. That's the idea, that's the sound amplitude of a female flying past the male. The black then is the male's antenna moving. And you'll see that at the point where you have that blue arrow, there's some sort of amplification happening. The signal, the amplitude of motion of that antenna gets higher, and it stays higher and stays higher and carries on being higher until you finally get to that red point at which it drops back off again. So there's this hysteresis. Increase the female sound. Eventually, at some point, you get the amplitude shifting up, as you can see in the bottom right graph. That amplitude stays high until as you drop the female sound until you get a point where it drops down. We're interested in where this actually comes from. We know that the mosquito antenna has motile mechanosensory neurons. And so the question was, were they the ones putting in the extra energy? So we opened up that the base part of the pedicel of that antenna where all the neurons sit, put an atomic force microscope cantilever down onto those cells, waggled the antenna with a PZT, also measured the electrical response of the cells as well, and saw what happened. And basically, so this is just one example, we were playing or putting in a stimulus with the PZT of 450 hertz, and you can see that the cells measured by the AFM are generally happily following that in a relatively linear manner. But then if you actually look at what's happening, you can also see a 900 hertz component appears as well. It doesn't appear straight away. You have to wait a bit until the amplitude of your stimulus is high enough. Then the 900 hertz component kicks in, carries on, and then stays on for a lot longer. So you get the same sort of hysteresis until eventually the input signal's low enough that then this one just drops away. And hopefully you can see from the fade in there that that's relatively similar to what we see on the external antenna. We're just seeing it on the sensory cells within the antenna system. So what's happening? Oh, sorry. So this graph shows red at the bottom. That's just the sound. Well, sorry, stimulus slowly increasing, nice sinusoid. And at some point, you start getting this second beating, you get that extra doubling frequency appearing within your signal on when you're measuring the cells there in the blue. Basically, the theory is it's a parametric resonance. It's an internal force being applied to the antenna to give it that extra gain. We know it has motor mechanical sensory neurons. It's pumping in extra energy in order to make that antenna move more. And of course, the most efficient way to do that is to use twice frequency forcing. And so the mosquito is using that mechanism to optimize its ability to amplify sound. So we have moth and mosquito, as well as humans, mammals, frogs, and various other insects, using some form of active feedback to enhance their signals. That's a very old graph there on the top right, but that's what happens on the human ear as well. We have motor mechanosensory neurons, which do a similar thing. They add energy into our hearing so that we can tune into particular frequencies, basically add some gain into the signal. Okay, if the insects do that, then can we look at a way of doing that with our microphones? And so we built a MEMS microphone which had both capacitive and piezoelectric transducers on it, as you can see there in the bottom right-hand side, to see whether we could do a similar thing to what you find in the moth and in the mosquito. So on the moth side, we were basically looking, can we change the frequency response? The obvious way to do that is by changing the stiffness of the system. So it's simply applying a DC voltage to some of the transducers on that MEMS microphone, and you can make the resonant frequency of that microphone run up and down. Of course, what you can then do is have some sort of automatic real-time feedback of that, so that you can actually make it follow a signal once you've picked up a signal. At the same time, we wanted to see, could you do a similar thing as we did with the mosquito? Now, this is more of a duty by cycle input. So this is basically putting extra little kicks of energy into each cycle or every other cycle in order to put more energy into that system. So on the top right there, you can see the linear version where there is no input of spikes, and you get the output linearly following the input that you've got there in blue. Turn on your feedback system, which is putting little spikes of energy into that. And you can basically, as you see in the red signal, do something similar to what you have with the mosquito system. And of course, because you're the one designing the feedback system, you can make it have the same hysteresis, gain, or attenuation. Of course, what you can also do then is combine those two together. So you can take your microphone, which as a passive system has a particular resonant frequency and a particular sensitivity, and then using these bio-inspired feedback mechanisms, you can make it change its resonant frequency. You can also make it change amplification or attenuation, such that you can move that microphone around in that space. But of course, then you can do that in a feedback manner such that it does it automatically. Okay, that's me done for today. And you can probably hear that my throat is beginning to give out a bit for talking about some of the work that we've done. I wanted to just flash one next slide, because of course, once we knew we could 3D print systems, then as usual in a lab, people ran off and started doing other stuff. And one of those other stuffs has been acoustic metamaterials. That's pretty much the subject of a whole other talk, but we've done 3D printing of Helmholtz resonators to give us nice attenuation band gaps. We've done 3D printing of ultrasound beam shifters, as you can see there in the middle, it's shifted the beam linearly. much larger systems where it's several different metamaterials piled one on top of the other, giving really wide band attenuation. Again, this is all at audio frequencies. And then much more recently, we've looked at basically tending towards what we would call active acoustic metamaterials, whereby because we can 3D print piezoelectric or magnetic systems, then we can actually change the resonance of our acoustic metamaterials. So you can actually make it do something different, give it a different band gap, for example. Looking at the future, well, there are many, many more insect ears. Hopefully people will carry on this work after I've retired. There are something hearing in insects, depending on which paper you read, evolved something like maybe up to 19 different times across the insects, in various different insects with all sorts of different ears. 3D printing of functional materials has really come on in the last 10 years since we started doing it. People are doing all sorts of interesting things with piezoelectric materials, which are more piezoelectric, much better conductive materials. Of course, the main problem is you need someone who understands materials and understands 3D printing in your lab to able to work out how to get the things to work together, but it's certainly getting there. Multimodal sensors is something which we're starting to look at. So you can imagine being able to 3D print something which isn't just a sensor. It might be a transducer, or it's not just a sensor for sound. Maybe it can switch to sense something else like airflow. And then, of course, tying that all into our work on active systems. And then finally, a little mention, just in case you know anyone who wants to do a PhD, we've just recently received funding from the Leverhulme Trust for the new doctoral school for nature-inspired acoustics. That's an interdisciplinary doctoral school where we've got people looking at nature-inspired materials, which will then feed into nature-inspired sensors, which will then feed into applications. We're particularly interested in sustainability in the built environment. And then that's actually tied into people looking at foresighting and future casting as to what you'd actually might use nature-inspired materials and sensors for. So some conclusions. Insect ears as inspiration, they can give you things like passive analysis of sound frequency, direction of what I've mainly talked about today. You can use active processing. Insects use active processing of the sound for gain or tuning. And so we've used 3D stereolithography as a way of exploring three-dimensional bio-inspired structures. Looking into the future, there are an awful lot of insect systems there to inspire us. And so we've started also looking at tying that together with how you can use active processing as a way, for example, of reducing DSP or mating a very simple sensor which can do one particular thing very well, but also very cheaply. And of course, because we've done all this development work on 3D printing, that's led into other acoustic stuff, acoustic metamaterials, multimodal sensors, etc. And I definitely need to use the word us. I've probably missed some people out of this. Hopefully I've covered in these names all the people who are involved in some way or other in the work that I've presented today. Main thanks go to Dr. Andrew Reed and Dr. Joseph Jackson, the academics who have been with me here at Strathclyde working on this, and before that for Joe. Dr. Roger Domingo Roca and Dr. Lara Diaz Garcia. Those are the two postdocs in the lab, but then there's a whole list of other postdocs over the last 10 plus years have been involved in the various different parts of this work. So Jeremy Jose, Roland, Yangsheng Botong, Brian, Cecilia, David Shearer, and Hannah, who was my first PhD student, who has looked with me at how locusts and moths here. Current PhD students who are working on this area and various different bits of 3D printing and making small devices. So Sam, Brendan, who's actually a biologist, Luke, Rachel, Dorothea, Paul, and Olawasian. And then the final list of mainly profs, the various different academics and colleagues who've inspired me over the years, Professor Daniel Robert and Professor Martin Gottfert, without whom I would have never got into insect hearing, James Fullard, who was a professor who looked at moths, who helped me with a lot of stuff, Andrew Mason, who's over in Canada looking at Ormia hearing, Fernando, who's down in Lincoln in England, who looks at all sorts of different insects. And I got involved with doing some very crazy experiments with Jerome, who did our original work looking at underwater insects, Michael, who we did work with on the moths, and then Jerome Casas, who we're currently collaborating with looking at the hair type sensors. And then finally, Dr. David Hardy, who has probably forgotten more acoustics knowledge than I will ever help to learn. So hopefully that gives us 10 minutes now just for some questions. I'll leave the share up at the moment. I've got one in the Q&A. Are mosquitoes auditory organs similar to those of bees, Johnson's organ on the antenna? I'll answer that live. The easy answer is yes, kind of. But only as much as it physically they are a bit different, but they are similar, yes, in terms of there's an antenna and then there's a sensory cell at the bottom. To say that a lot of insects basically have that sort of structure, to tell a slightly funny story, way back when, given that we're in Glasgow, we thought it would be an interesting idea to look and see whether midges could hear sound. And the simple answer is, after quite a bit of work by a couple of students, midges don't appear to hear sound at all, or at least if they do, we could never prove it. And we've also found that going way back in the past with insects. You know, often it looks like they must, you know, we've even seen insects that generate sound, really loud sound. They have structures which look like ears, but the actual really difficult part of the biology side is proving that they're actually doing anything with it, unless there's a very obvious reason like mating or trying to avoid predators. Then it can be difficult to prove they actually can hearing. Yes, there will be a recording of this. It is hopefully been recording, and so it would then be that recording will be put up on the UCAN website along with the other webinars. Okay, any other more questions for the Q&A? Not a question. Okay, well, thanks very much for saying that. I've always, I mean, to give a little bit of the past, you know, I came into a biology lab as an engineer who had studied biology at GCSE level, started asking questions of the biologists to then very quickly work out that they actually didn't know a lot of the answers, because of course, you know, biology is not an easy thing to work on and tends to need a lot more funding than is available. So I immediately found it far more interesting to get involved in the actual biological stuff. And it's only over the years since then that I've tilted back to the engineering. Okay, I'm going to stop the share there.